evening, everyone, and welcome to this webinar on the pharmacology and clinical application of CBD, brought to you by M2Bio Sciences. This is our first webinar, and we hope to bring more content like this to you in future. In recent years, CBD has become a subject of scientific inquiry and medical interest, compelling healthcare professionals worldwide to delve into the intricacies of its chemical compounds and the profound effects it exerts on human physiology. This webinar aims to equip healthcare professionals with a holistic and better understanding of CBD's pharmacology and clinical application. Some general housekeeping. Please post all questions you have for the speakers in the Q&A box, which will be answered at the end of the two presentations. For all technical queries, please post these in the chat box and our support team will assist you. If you are unable to view the whole webinar, a recording will be made available in the next 2 to 3 days. Healthcare professionals should please provide their professional registration numbers so that we can submit your CBD point to the HPCSA on your behalf. So to introduce our speakers for this evening, our first speaker is Prof Vinesh Maharaj. Prof Maharaj is the director of the Biodiscovery Centre in the Department of Chemistry and is the Deputy Dean of Research and Postgraduate Education of the Faculty of Natural and Agricultural Sciences at the University of Pretoria. He is a natural product chemist and is trained in the discovery and development of new drug leads based on biodiversity samples. His main aim is to create a unique repository of natural products comprising 10,000 plant samples, which forms the basis for the Natural Product Library, the first of its kind in Africa. He is also actively involved in the development of herbal medicines based on his experience he gained working with Pfizer, Phytopharm, and Unilever. Prof Maharaj heads up the M2Bio-Discovery Centre for AI-Driven Phytomedicine Research at the University of Pretoria, where research is being conducted on behalf of M2Bio Sciences. Our second speaker is Dr. Skye Scott, who is a general practitioner. Dr. Scott has a special interest in integrative medicine, collective well-being, and reversing chronic diseases of lifestyle with behavior change. Skye is a co-founder of Health with Heart, which brings high-quality, holistic occupational health and wellness to large corporations. She hosts a podcast that explores ideas around individual and collective well-being. Her passion for community was first stirred while doing her community service at Kladi Clinic in Soweto, where a soup kitchen that she started still feeds chronically ill patients during the week. And our last guest is Zeev Crane, who will be facilitating the Q&A. Zeev Crane is the current section commander of the South African Narcotics Enforcement Bureau at the DPCI, or the Hawks. He coordinates drug matters for both national and transnational activities. His role encompasses a diverse range from legislative inputs and coordination of investigations to scientific guidance in forensic, analytical, and medicinal chemistry, collection and reporting of drug data to the implementation of cutting-edge technology. He has a chemistry, computer science, and law background with a BCom in information systems, a BSc in chemistry, honors in chemistry, masters in medicinal and or organic chemistry, and an LLB. So a very colorful background there. He has now also been accepted to undertake his PhD in the area of AI applications for natural product drug discovery under Prof Maharaj at the University of Pretoria. He also serves as the Chief Scientific Officer for M2Bio Sciences. So without further ado, I would like to please invite Prof Maharaj to begin.
Thank you, Nicola, for a very comprehensive background. I see you've dug deep into my past as well. So thank you for the introductions. I'm just going to try and get to my video. I think you should be able to see me now. Good. Yes. Just allow me a second to share my screen. Perfect. Okay, good. So good evening, colleagues, and thank you for giving me this opportunity actually to share with you something that actually has been very active in the past few years, really around CBD. And my presentation really focuses on the chemical compounds of CBD, and the main reason of that is these compounds have a direct effect on the pharmacology and then the eventual clinical observations and clinical studies that actually arising from the pharmacology. And these compounds are very specific in the way they act. So I thought it'd be very appropriate to actually provide you more background to what are these compounds that are present in cannabis, and then how they act at a pharmacological level. So as Nicola says, is that I have quite a few hats, but one of the most recent hats is that I'm actually the M2Bio Research Chair in collaboration with M2Bio Sciences and the University of Pretoria. So just some background and some history to the medicinal use of cannabis, and actually dates back quite a long time, back down to the 1500s before BC. And at that time, in fact, there were records on papyrus on the use of cannabis as an anti-inflammatory uses. So dates back quite a long time, 50 AD, by De Coroidus actually mentions the cannabis is used in one of his writings. It's also used medicinally, and extensive descriptions more in the 1800s. Moreau is actually was a pharmacist, writes about the psychosis induced by uses by of cannabis. Medical use of cannabis by Queen Victoria and the Empress Elizabeth of Austria is also recorded around the 1800s as well. But it was only around 1940 that the actual chemical compound called CBD that was identified by Roger Adams, and the structure was determined in 1963. And that's the CBD that was first identified and its chemical structure then produced. THC was then identified a bit later, and that was actually done. That was going to move. Sure. That was done in 1964, where THC was identified by Raphael McCollum, and that was in Israel. And that was some groundbreaking work that actually was done to identify the THC compounds. So those are two main compounds that was discovered more in the 1940s and the 1960s. So some background as to the source of cannabis. So cannabis comes from one of two plants. The first is actually Cannabis sativa. Sativa simply means cultivated and describes a common hemp plant. So Cannabis sativa is the common hemp plant that was widely grown across Europe in the Linnaeus time. It was meant to produce mainly CBD. So sativa is known to be producing mainly the CBD compound. The second species is Cannabis indica, which originated from India. And after the origin of the first sample of this highly psychoactive plant that had reached Europe. Now, Cannabis indica is meant to produce mainly THC. However, over time, due to the supply and demand, farmers actually started to grow both the species. And both the species, and they use the seeds intermittently. And then at a point in time, you could not distinguish between the two. And because of open pollination as well, there became several hybrids between the two species. So in terms of actually getting a cultivar to produce just the CBD or the THC has become a bit of a challenge. And nowadays it actually has to be controlled by actually farmers themselves, who actually have to be knowing exactly which species they are producing and actually control the way they're actually cultivated. So they actually know whether you're actually having a CBD-rich cultivar or a THC-rich cultivar. And that's the way it actually is done now. And it's quite a controlled process as well. So cultivation plays a critical role in the supply for a specific type of cannabis compound. Just some basic terms and definitions. So cannabinoids is a class of chemical compounds that act on the cannabinoid receptors, and they are the active constituents of cannabis. So there are many cannabinoids, and I think there's well over 100, and I most recently read that it's like close to 140 cannabinoids that have been isolated and identified from the cannabis plant. But the two most important ingredients is tetrahydrocannabinol, THC, and this is a psychoactive cannabinoid responsible for giving the so-called high. I'm just going to try and get my laser pointer. Good. The second compound is cannabidiol, CBD, and this is regarded as the non-psychoactive cannabinoid. And if you look at the two chemical structures, interestingly, they have exactly the same formula, so very difficult to distinguish between the two using some of the techniques that we have. But it's a simple bridge between these two rings, and actually closes the rings and produces the cyclic form. Whereas your CBD, this ring is not formed and actually is an open form. So very closely related, the two compounds, but the effects actually are quite, the difference in the effects is quite pronounced. So as I said, there were several compounds that are produced by cannabis, and close to 100. And they actually have very minor differences between them. I mean, some of the very basic differences, if you look at THC, has a certain number of carbon atoms, whereas, sorry, that's THC. And then if you look at another derivative of it, it has a bit more carbon atoms there as well. And some structural variations, and the same with the CBD as well. You just vary the side chain or the alkyl chain on the side there, and then you have a different compound. And in fact, more recent work is actually looking at the effect of these individual compounds rather than just the main THC or CBD. So a lot of research is actually going on recently is to look at how these other compounds also work, and pharmacological effects of these, or even sometimes actually the synergistic effects that when they work and give these pharmacological effects. How is it produced? So the production of CBD is typically an extraction process, and this is basically either a cold press producing an oil, or a carbon dioxide. This is actually supercritical carbon dioxide extraction, and it's actually quite a neat process because it gives you the advantage of an extract which is actually solvent free, whereas you also can make a solvent extract using solvents such as propanes, such as pentane, and even ethanol is the most commonly used one. But in these cases, you actually have a solvent residue in the end product. The extract that's produced using these extraction techniques is called a full spectrum CBD. The second one is basically a distillation process, and it typically takes a full spectrum CBD that moves into what you call a purification process, what we call short part distillation or thin film distillation. And this process actually removes any THC and removes and gets it to what you call trace levels. So this is a broad spectrum CBD, which is mainly CBD and almost trace quantities of THC. And finally, if you then take this broad spectrum CBD, and through a process of winterization, which is really adding some alcohol and keeping it at sub-zero temperatures, produces a high purity CBD through crystallization. And that is predominantly CBD. So if you look at the full spectrum CBD, this is the first process. It typically contains legal amounts of THC, typically about 0.3% of THC. That's more or less over there. It requires much less processing in the broad spectrum, and is predominantly CBD, and of course still has some of the other minor constituents of the CBD analogs or the other minor compounds. So that is the broad spectrum or the full spectrum CBD. It's ideally for those who are open to THC and seeking the so-called enhanced effect. If we move on to the broad spectrum CBD, this contains all the cannabis plant compounds, contains no or trace quantities of THC. So this is predominantly CBD and some of the minor compounds of the CBD class. And generally we call this the CBD hemp oil. It is minimally processed and can be used in states where cannabis is legal. Perfect if you're subjected to drug tests as well. So that's the profile of the cannabis, broad spectrum cannabis. And if we move on to the final one, which is the CBD isolate, and this is the highest quality CBD. It contains no THC, so there are no concerns about any psychoactive effects. It will not test positive for THC in a drug test. It's odorless and tasteless. Can be added to any beverage or food because of its odorless and tasteless. And if you look at the profile again, it is mainly CBD with just some minor trace quantities of the other cannabinoids. So this is the highest purity CBD isolate. All right, so quality control becomes critical for this because of the varying concentrations of the CBD and the THC in the different types of extracts that are produced. The regulatory environment does require one to actually be able to distinguish the concentrations of THC and CBD. So the most common techniques, other than the other inspection and other quality control measures that are required, such as microbial contamination and so forth, the two most common techniques that are used is gas chromatography, mass spectrometry, and this process typically takes your sample, puts it through a gas chamber, and because of the volatility of the different compounds, they come out at different times. And then we have a detector that's able to detect each of the different compounds at different times. The same process with HPLC mass spectrometry. HPLC is just high performance liquid chromatography, so it's not gaseous, it's liquid, and gives you the same end result, which detects each of the compounds separately. And one is then able to provide the final concentrations of the individual compounds in the extracts. So whether it's milligrams per kilograms or milligrams per liters, it can give you that final concentration. So that's individually for CBD and THC. So HPLC mass spectrometry is just gives you a bigger picture and a better picture of the samples, because it doesn't rely on just volatile compounds, but all types of compounds. Right, if we just look at the South African landscape in terms of the regulatory environment of SAHPRA. So it's regulated by SAHPRA under the Medicines and Related Substance Act of 1965. But as of 23rd May 2019, one of the main regulations, the regulations kind of were amended. And that says that no prescriptions are required for Schedule 0. And in terms of cannabis, processed raw cannabis products with less than 0.0075 CBD and in only natural quantity cannabinoids are present, and these are the full spectrum CBD as an ingredient. So these can be used in a zero scheduled cannabis product. On the complementary medicine side, and that is also then, and it's almost more over-the-counter type of medicines, a maximum daily dose of 20 milligrams CBD. Also, in terms of the full sales pack, a total of less than 600 milligrams. So 20 milligrams of CBD and less than 600 milligrams of the total pack for the broad spectrum as an ingredient. So these are the over-the-counter and almost Schedule 0 type of CBD products that are permitted to be traded under SAHPRA. Under the pharmaceutical grade pure CBD, it is rated as a Schedule 4 medication. And in this case, the THC is also rated as a Schedule 6 medication, unless THC makes up less than 0.2% of raw processed plant material. So if you have raw plant material, it has to have less than 0.2%. And if you have THC, it's got to be less than 0.001% THC in a Schedule 6 medication. So these are the criteria by SAHPRA if you actually are being able to produce any of these products and then be able to trade in it as well. All right, so let's move on in terms of some of the biology of the cannabinoids. Let's just briefly talk about the absorption, distribution, metabolism, and excretion. So what is the fate of CBD as it actually enters the body? So administration could be oral, sublingual, buccal, topical, or by inhalation as well. So these are the common administration routes of CBD. So in terms of distribution and its bioavailability, we find that the highest bioavailability is by inhalation, followed by buccal or sublingual, and finally the least bioavailable is the oral bioavailability. We find that CBD is rapid systemic distribution to the vascularized tissue, that's the heart, brain, lungs, and liver, and diffusion of accumulated CBD from adipose tissue. and chronic exposure to cannabinoids lead to a persistent pharmacological effect. In terms of its metabolism, of course, this is taken orally. So it basically has the hepatic first pass metabolism if administered orally. This is avoided if you're taking it through buccal and sublingual or through inhalation, and even transdermal routes. So the primary metabolite that is produced through metabolism in the liver is called 7-hydroxy-CBD. So it's converted to 7-hydroxy-CBD and excreted in that form. So then further on, there's some non-hepatic metabolism that also occurs in the small intestines and the brain. CBD metabolite, the bioactivity is still largely unknown. That's a 7-hydroxy-CBD, so it's not fully understood. That's what its bioactivity is. In terms of elimination, the extended elimination half-life or excretion is between 1 to 1.5 days. Okay, so now we move into some of the pharmacology of the cannabinoids, and here we relate to the endocannabinoid system. So the endocannabinoid system is really a system that the body has to maintain homeostasis. So it really consists of an integrated process of endocannabinoids, enzymes, and proteins, and your two main receptors called the cannabinoid receptors CB1 and CB2. And when there's an imbalance in the body, the body then generates endocannabinoids, which through the production of through the use of some enzymes and proteins, and then these then interact with the cannabinoid receptors, and through the cannabinoid receptors then actually provide some pharmacological effect, which then brings back the body to a state of balance and brings back normality. So this is the endocannabinoid, a very, very broad description of how the endocannabinoid system actually works. So we just move on in terms of some of the endocannabinoids that are produced by the body. So when there's a response and the endocannabinoid system needs to be active, the two main endocannabinoids are 2-arachidinyl glycerol, or also called 2-AG, and then anandamide, and that's also called ANA. So these are the two main endocannabinoids which bind to the receptors, the CB1 and CB2 receptors, and also use the proteins actually to create this homeostasis, and that's how basically the body continues maintaining its balance through the endocannabinoid system. So these are the two naturally produced endocannabinoids. There are others as well, but they're not as active as the two. All right, where do we find the cannabinoid receptors? So the cannabinoid receptors are found in several parts of the body, but the main part of the body is actually the brain. You'll find the CB1 receptors are primarily in the basal ganglia, the prefrontal conchus, the hippocampus. So it is actually found predominantly in the brain, while other parts of the body it's also found in, I think, in the spleen as well. While if you find so the reproductive organs, the muscles, vascular system, and the lungs, while the CB2 receptors are found in the colon, the spleen, and the immune system. So that's quite important actually occurring in the immune system because it has an effect in the immune system. So these are where primarily where the CB1 and CB2 receptors are located in the body. Right, so just moving in terms of how the mechanism of action actually works. So typically the CB1 receptor, that's the CB1 receptor, and it has what you call a binding site. And the binding site is then open to what you call an agonist. And once an agonist then binds to the receptor, typically an agonist like we see in this case would bind to the receptor, it then signals a cascade of reactions, and then you have a full pharmacological response. That's typically when an agonist binds to a receptor. In this case, the CB1 receptor, what we observe in this case here. So you also have what you call a partial agonist binding, and this is classically THC. So THC is an agonist binding ligand. So it binds to the receptor in what we call partial way, but it still does cause a cascade of reactions which then results in what you call a partial pharmacological response. So this is the way THC actually works, binding to the CB1 receptor. An antagonist is something that actually blocks the receptor site and prevents the agonist from binding to it. So it actually stops the agonist from binding to it. And when it stops that, you do not get the pharmacological response. So in the CB2 receptor, what we see is this how CBD is how CBD actually works. So what CBD works in the way CBD works, CB2 CBD binds to the CB2 receptor as in an antagonist way or inverse agonist. And that way it prevents THC from binding to it. Whereas in the CB1 receptor, CBD actually binds in an unusual way, right? It binds in what we call an allosteric modulator. So it's not binding to the pocket or the site. It binds to an allosteric way. Even though THC may still bind, CBD binds in an allosteric way. And when it does this, it inverses the effects of the THC. And that is why CBD has the potential to treat central nervous and peripheral disorders while avoiding adverse effects. It is mainly because of this binding in an allosteric way. So not on the pocket, but it binds in a different way inside the cellular membranes. And that is actually quite unique. And that's what makes CBD very, very effective in actually producing pharmacological effects on the nervous system without any of the side effects. So CBD does not only act on the CB1 and CB2 receptors through the agonist or the allosteric ways, but it also binds to several other different targets, either as an agonist or regulating these as well. And in fact, it actually works on CB1 and CB2 in a way by downregulating by ensuring these are not degraded through what you call an FAAH enzyme modulation system. So it ensures homeostasis by producing the two CB1 and CB2 receptors to ensure that the AMA can actually still bind to these as well. In addition to that, it also binds to 5-HT, TRPV1, D2, GRP55, and MOR, and many of these are then recorded, have pharmacological effects that actually regulate things like epilepsy, pain, even opioid addiction, and that's your MOR, and your sodium and potassium channels that it actually works on as well. So CBD reacts on many other pharmacological targets other than the CBD1, and these actually provide many more of the other pharmacological effects that we are starting to see as well. So because of all these pharmacological effects and the different targets it actually works, these are typically the end effect, medicinal effect we actually see. And that's improvement of sleep, reduction of inflammation, and in reduction of inflammation is directly related to CBD working on the immune system. And because it works in the immune system, it results in the production of cytokines, and that is used for the treatment of inflammation and, in fact, also cancer as well. So it helps with chronic pain, relieves anxiety, can even stop bacterial growth, control blood sugar levels, and also I cannot see my screen over here, but yes, helps with some intestinal problems as well. So that's typically your CBD oil types of benefits that we are starting to see as well. And more on the medicinal uses as well. CBD is now being implicated to be used or been tested to be used in various diseases, life-threatening diseases such as cancer, Alzheimer's disease, depression, epilepsy, anxiety, and also helps with body weight and sleep issues as well, including some of the schizophrenia as well. So it's actually moving into quite a vast number of medicinal uses as well. Okay, I think that's my last slide, but just to maybe share with you something that in the old days, it was quite different at that time. You could actually buy heroin at that time over the counter. You could buy cocaine in drops for toothache. You could buy opium as well. You could buy cocoa wine with some cocaine in it as well. Vapor oil with some opium as well. So yes, it was quite different in the older days. But nevertheless, these are medicines that are still used. And with that, I'd like to thank you for your attention.
Thank you, Prof, for that amazing beginning to this evening's talk. Give me a second while I just share my screen. Nicola, can you see my screen?
We can see your slides, yes. Great.
Great. So I'm a everyday GP, and I'm glad to be in a room with so many academics so that they can answer any questions or put their hands up if I put a foot wrong. But I think what's unique about my what I bring to the table here is that I engage with patients on a daily basis who often meet the limitations of pharmaceuticals that are legally available to them. And the pharmaceuticals are so often at a price, which you'll see some of in my presentation. So I'm not going to give you another history lesson, but I wanted to just shine a little bit of light on the way that so much of the language is framed in a lot of the studies that I read on the use of CBD and THC in preparation for this talk. And if I think about the large array of drugs that we are able to script on a daily basis, especially for psychiatric disease, many of which can cause sudden death. A little bit of psychosis once in a while is not such a bad thing. And so it's almost frustrating to see how slow sometimes bringing, as you can see, ancient healing medicines to the table can be. I'm not going to spend any more time on that slide. And maybe just to remind you again, Prof alluded to anandamide and 2-AG, which are your endogenous cannabinoids. And these are so integral to the development of our brains in utero. We know that the levels found in fetal circulation of these compounds are much higher than in fully grown humans, and they are very instrumental in neuroplasticity, the outgrowths and embryology of the brain and the central nervous system. And as you have seen already, can be instrumental with appetite regulation, digestion, your gut-brain axis, how you experience the world, your anxiety levels, your emotional experience, cognition, and memory. And I've got a picture of a few jumping, skipping folk over here, because the best way to upregulate your own endogenous production of these compounds is through exercise. So if you aren't already doing that, please begin. Prof has already alluded to this. I think what was really exciting and interesting for me to learn about these compounds is the diverse array of applications that they have, and that they are not psychoactive. And so being able to prescribe something that's not going to put your patient in an altered state, but is going to have all of these positive effects without any of the negative effects that so many of the drugs that we have at hand provide, is really a revelation. I'm going to skip over that. And so just to reinforce again, in the brain, the potential for therapeutic intervention is around synaptic transmission and motor control. We see this in multiple sclerosis, for pain and anxiety and sleep. And then in the context of psychiatric disease, for anxiety, depression, and sleep disorders. And then, like Prof said, homeostasis, which sounds like such a vast and untangible concept, but your endocannabinoid system is so vital to keeping your body in check. And then outside of your brain, your autonomic nervous system, your immune system, these compounds often have an immunosuppressant effect, which is often what we are looking for in autoimmune disease and in some cancers. And so the immune modulating effect of these drugs or potential drugs is really exciting. And then in inflammatory conditions, the biggest global burden that we're all facing at the moment is obesity and diabetes, and the microvascular consequences of those disease processes. And there's certainly hope that some of these interventions may be able to support us out of that problem. And then foods that naturally contain cannabinoids are listed here, and that's something that you can also just spice up your diet with for elevated mood and better homeostasis, which I think we're all after. So this slide gives you a little bit of a summary of what FDA drugs are currently approved and in use that are derived from cannabis. The first one is a THC formulation, which is an appetite stimulant and an antiemetic, and this is approved for use in advanced HIV with wasting, anorexia, and vomiting, and in chemotherapy-induced nausea and vomiting. And then Sativex is a combination of THC and CBD that is used for the treatment of pain or spasticity in multiple sclerosis. And there's that motor effect that we spoke about earlier. And then there's a THC mimic called nabilone, which is used for the non-motor symptoms of Parkinson's disease. And then the one that has probably got the most airtime in the press is for use in some rare forms of pediatric epilepsy. And this is an oral solution of CBD called Epidiolex. And this, in many children, has completely stopped to what were previously intractable seizures, or significantly reduced the amount of time these children are seizing in a day, which is really transformative and hopeful. What are the risks associated with cannabis? I know I spoke about the bias around framing in the beginning of this talk, and certainly every single drug and every single thing on the planet has a shadow, and so certainly THC and CBD have these too. I would exercise caution in pregnancy and breastfeeding, especially because of the role of the endocannabinoid system in the development of a small fetus. And so I think even the use of CBD in pregnancy, even though this might appear innocuous with it being included in certain foods and over-the-counter sweets and that sort of thing, should be avoided for pregnant or breastfeeding moms. We know that these compounds are really lipophilic, and so they cross the placenta. The placenta itself actually has cannabinoid receptors, and so there might even be a secondary effect on the fetus via the placenta, in contrast to the direct effect on the endocannabinoid system of the developing embryo. And then in adolescence, there are so many studies. I had to draw the line at stopping to read in preparation for this talk, because as any of you will know, you can find for and against arguments for everything in medicine, and it can get quite muddy and confusing. But there does seem to be a general consensus that significant smoking of cannabis twice a week or more can induce psychosis and increase your psychotic risk. And this is especially important in adolescents and young adults between 16 and 25. So I'm going to take you through, I've just picked a couple of studies that I found really interesting. And as I've already mentioned, I couldn't find anything really definitive. Doctors love protocols. We love to know, in this situation, do this, and in this situation, do that. And unfortunately, the research is just not there yet, but it is rapidly catching up. And I think because many of these compounds are now available to lay people, as Prof alluded, people are beginning to create their own anecdotal evidence and experiment with these things where pharmaceutically available products are not meeting their needs. So chronic pain affects up to a third of the world's population, and only 11% of that population have relief from the drugs that are available to them. We know that we have a massive opioid crisis in the States, and we know that a lot of our drugs that are available for pain cause significant alterations in gut function and can compromise renal function. So CBD can be really effective in treating pain. This was a retrospective study done by a Canadian clinic that uses CBD specifically for pain, and they selected 279 patients out of 1,100 based on the criteria to enter the study, and they were all offered a really CBD-rich treatment. And those who had moderate to severe symptoms had an enormous reduction in pain, anxiety, as well as depressive symptoms, whereas those who had really mild symptoms, the effect was not particularly noticeable. So this is really hopeful because, as Prof alluded, the side effect profile of these drugs is really safe in the right patient. This was an experiment using full spectrum hemp oil, which is predominantly CBD, on a whole host of poor mice. And the way that they injured the mice or created a neuropathic pain was to damage their trigeminal nerve. All of us have a trigeminal nerve too that runs across our face. It has 3 arms. And so they injured the trigeminal nerve, and they tormented these poor mice. In the one cohort, and in the other cohort, they administered full spectrum hemp oil, and there was an 11-fold reduction in perceived pain by these rodents, which is really significant. This study compared CBDA, which is one of the compounds that Prof mentioned earlier, to COX-2 inhibitors, and the effect of these drugs was almost equivocal. The challenge with COX-2 inhibitors and COX-1 inhibitors, these are both under the arc of anti-inflammatory drugs, for those who might not be familiar with them, is that they can affect your gut, they can affect your gastric lining, and they can negatively influence your kidney function. And as you saw from the metabolism of these drugs in Prof's introduction, that the kidney wasn't really mentioned. In fact, I read some studies where use of CBD seems to regain some renal function in patients with chronic renal disease. And so the application for inflammatory pain is really powerful, and the evidence is really strong. This study was the only one I could find with some dosing recommendations, and they recommended a CBD-centric approach. What was interesting about that retrospective Canadian bunch was that they started everybody on this full-spectrum hemp oil, and then in phase 2, they added small amounts of THC, and they didn't notice a significant impact with the addition of THC, which I found quite surprising. So this study recommends starting with CBD at doses of 2.5 to 5 milligrams once or twice per day, and increasing the dose every 2 to 3 days, so titrating to effect, and with the potential to add a THC component once you've reached 40 milligrams of CBD if you haven't attained pain control. And caution is mentioned in all of these studies in the very young and in the very old. Medical cannabis and its constituents in pediatric, developmental, behavioral, and mental disorders. This is really interesting, and there's a lot of research in autism spectrum disorder, which is so much more prevalent today than it has ever been. If you look at the graphs for this currently, it's really staggering, and I wish I knew all the answers to exactly why. But in silent children or antisocial children, these medicines have proven to be really transformative. I know this anecdotally from colleagues, and unfortunately all of these studies that I could find had really small sample sizes, but I expect that parents who have tried everything are moving on to this sort of thing and could probably give us a lot of valuable insight. This was an interesting study about alcohol use disorder, and what this study explored was that the CBD and other cannabinoids had a counter effect on the microbiome and the gut-brain axis to the effects that alcohol have. We are, the term leaky gut used to be something that only the pendulum swingers used, but it has crept into the current allopathic literature. And I think it's integral in understanding some of the autoimmunity that we are seeing, and is probably driven by our industrialized food production and the refined sugars and carbohydrates that so many people are being sustained on. And so when you have damage to your microbiome, you get damage to the tight junctions of that single cell gastrointestinal lining, and then you have all sorts of molecules crossing that barrier with a ripple effect on your immune system. And the use of cannabinoids modulated this effect in this study, and reduced the craving and use of alcohol in people with alcohol use disorder. So I think anything that's going to speak to the microbiome is going to speak to every organ system in the body, because the microbiome is so integral to every organ system in the body. And so I think this is a really exciting area of research to watch. Immune function, we've mentioned a couple of times. It has an immunosuppressant effect, and that's why it's also really important not to use while pregnant, because we don't want an immunosuppressant effect on a fetus. But you get modulation of T and B lymphocytes, which are so important and central to so many disease processes. You get reduction of degranulation and chemotaxis of neutrophils, and the behavior of macrophages also changes when we stimulate the endocannabinoid system. And so there is really exciting potential for use in autoimmunity. And I really look forward to being able to have these tools available to us, because so many of the drugs that we do have available have really awful side effect profiles for patients. And then this is my last slide. I thought that this was really interesting. And I know it's not about CBD, but we're in the arena. Smoking THC for hypoactive sexual desire disorder. And this had a divergent impact on different people in the study. And this is something that I think maybe we don't talk about enough, but in couples who have discordance in this arena, or for people who are who this is affecting their life adversely, a large proportion of people had so there were two groups. The one group had an increase in prolactin after the inhalation, and didn't have increased sexual desire. And the other group had a drop in prolactin, which usually has a reciprocal raise in dopamine, which is, as you know, the neurotransmitter that drives us to do the things we want to do, or we think we want to do. And I thought this was really interesting and also something to watch. So that was my last slide. I hope that you learned something and that you found it interesting. And Prof, I hope I didn't say anything that wasn't true. And I'm going to hand over to Zev now.
Hi, everyone. Nicola, I don't know if you can allow my video, but I can also stay in this mysterious state. But anyway, let me thank Dr. Scott and Prof Maharaj for explaining really technical concepts in a way that we can all understand. I know there's a lot to take in. This was high impact information, and even for a scientist, I think I'm going to have to watch this recording a number of times so that I don't miss anything. But really, it was high impact, and I thank you for that.
Sorry, can I just interrupt? I think you should be able to turn on your camera now.
All right.
Just have a look.
Great,
yeah.
Excellent. Hi, everyone. Yeah, just as an aside, I actually got to meet Raphael Mechoulam, the Israeli scientist who isolated THC, and actually begged him to do my PhD under him, but he was already well into his 90s. So I'm glad that I get now the opportunity to continue the research under Professor Maharaj. It's very exciting. Okay, so let's see. Now we can go to questions and answers. I just want to see if there's any questions available. Okay, so Dr. Scott, or may just be informal, Sky. We were speaking about, I know you were speaking about dosages of CBD in terms of pain relief but just in terms of general, I think this is the question that is on everyone's mind, especially when you're seeing CBD and infused in so many products, and like you said, in sweets. And what is, in terms of general dosages, just for proactive, what sort of dosages should we be looking at in terms of CBD consumption?
Thanks, Zev. So to be honest with you, I couldn't find anything for just optimal vitality in the research that I did. And like Vinesh mentioned, there's a 20 milligram per serving regulation from SAPRA. One of the people that I did talk to in preparing for this suggested that 33 milligrams was optimal for taking the edge off of daily stress and sort of giving you a rejuvenated zest for life. But perhaps Prof can weigh in here.
Prof,
Okay,
if you-
I'm going to talk behind my screen. I don't think you can see me, but yeah. So I think, as you say, it's all dose related, and working out the exact dosage has in certain instances, in many cases, still has to be done. So often that is actually done in clinical studies to determine the effective dose. And much of that work is actually going on extensively around the world. So if you turn back and you look back, it's because of the history of the use of cannabis. Much of this work was not done, and it's only now recently that people have realized, and the research has shown that the benefits of CBD are far beyond this what we experience of THC. The interest has now been is to can we actually do the research and actually come up with CBD with actually the right dosage. So a lot of the work is actually going on. Critical to that, of course, is has to do with quality. So I would imagine that what is very important is the manufacturer or whoever is actually selling the product has to ensure that the right concentration of CBD is available. It's on the package because, as I showed in my presentation, that you've got to determine the concentration of your CBD in milligrams per kg or whatever that is. And if that is not done, you cannot translate that into a dosage. And I would think that that is a critical part of actually for whoever's manufacturing and producing and selling to ensure that the correct dosage is translated from your ingredient into the formulation.
Yeah, I think it is true. I think it was very sad that for a number of decades research was actually shut down, and we're now entering a really incredible era where we can once again begin this research, particularly around things like dosage and also sort of resolving between the different cannabinoid analogs. Let's just see what other questions are here.
Some of the studies that I saw on autism use, on autism spectrum disorder, we're using 20 milligrams per kilogram per day. So, I mean, that's for an average adult 60 times, 60 of these servings that you've mentioned. And so I think it's really variable. But what I also think, I think with any drug, I think we're trained pharmaceutically to think that everybody needs 10 milligrams or 20 milligrams. And I think our biochemistry differs. And what's nice about these formulations is that you can titrate to effect, which you can't do with so many other pharmaceutical preparations. And so I think that while we're waiting for more evidence and direction, start low and go slow is probably a good dictum to go by. But I think that the question was specifically around immunosuppressant effects, and I think you need much higher doses for that effect to be.
Can I weigh in here a little bit?
Yeah, please.
So there was a practical guide. I wonder if I could turn on my camera. Oh, sorry, I don't know why. The gremlins. Yeah, so there was a practical clinical guide that was published in 2019, and I can share this with the participants, where a bunch of doctors came together to sort of write a protocol for people to use. And they cited a couple of studies in there. And I know when it comes to epilepsy, they use really high doses. I think it was up to 1,200 to 1,500 milligrams per day. And I know that was used safely for about 4 weeks in those clinical trials. But because CBD is metabolized in the liver, it's really important to do liver function tests beforehand and then monitor the liver function over time, just to make sure that everything is functioning properly because of how CBD is metabolized. So that's obviously extreme, but I do know other studies, there were lower amounts. I think it was around 600 milligrams per day for about 6 months that they used it safely. So we definitely need to do biochemistry. And as healthcare professionals, we have to assist patients through this journey. We're there to guide them, and having a practical guide like that one that was published in 2019 at least gives us a starting point to work from.
Great, thank you. Thanks, Nicola. See here, Bianca's got a question. How high is the impact of metabolism on doses of CBD? I think with the assumption that quality is even across the board. Dr. Scott, I don't know if you want to take this one.
I'm not sure if I understand the question. Is the question about what is safe to metabolize, or when you're talking about metabolism, which metabolism are you speaking about?
Yeah, I'm also trying to interpret the question. I wonder if so let me just read it again. How high is the impact of metabolism on doses of CBD?
Nicola just mentioned that you need to be mindful of liver function. So maybe you're talking about safety of higher doses. As I mentioned a little bit, it seems to be quite safe from a renal point of view. Zev,
right.
Are we on the right track?
Yeah, I think it was answered.
Okay. Cool.
Yeah, I mean, maybe the other question is also, I think with all compounds they have a half-life, if in the body, or pharmacological effect. I mean, Dr. Scott, do we speak about it as half-lives in terms of pharmacology?
Yes, absolutely.
Right.
We do.
And do we have what is the general half-life for CBD?
That was definitely on one of Prof's slides at the bottom in red. I just can't remember what the number was.
Prof, you want to take this? There we go.
It was 1.5 days.
So metabolism obviously is all mainly by oral administration or oral dosage. And of course, as per your earlier question as well, in terms of the dosage, because your formulation plays a critical role as well into the bioavailability. So depending on what your formulation, like a liquid formulation, is more bioavailable than maybe a solid formulation, like in a tablet. So that also plays a role in terms of absorption and metabolism. And so ultimately, metabolism in this case it's converted. CBD is converted to 7-hydroxy CBD, which is the excreted form after metabolism. And the half-life or the complete excretion is 1 to 1.5 days.
Right. I guess there's two elements to it, right? One is how fast do we see an effect once we ingest it or take it into our bodies, and then how long does that effect last? I guess so that there's sort of a dual element there. To the Prof, I want to ask a question just in terms of, I mean, growing up, I always used to hear sativa, indica, and the different effects of sativa and indica. In terms of the analysis, I know that the gold standard you were speaking of is GCMS analysis, which separates and isolates and identifies the different molecules. Can we see a clear distinction in the GCMS or analysis in terms of separating between a sativa and indica strain?
Probably not between the strains, because these are so complex with so many other chemical or cannabinoids present in them as well. But classically, you'd expect to see a sativa strain that's rich in CBD and indica strain rich with THC. In that way, if you look at the either the GC or the HPLC mass spectrometry, you would see a concentration-dependent difference between the two species. So you should see much higher THC in the indica strain as compared to the sativa.
Thank you, Dr. Scott. I think we mentioned the enhanced effects if you're taking something with let's say a significant amount of THC, right? That's sort of now you're going to the psychoactive where you really say consciously and cognitively feel the effects. So whereas CBD, I guess, is you don't really experience that sort of conscious psychoactive effect. With THC, you certainly do, and that experience can be quite unfamiliar for certain people. So I know in your clinical experience, do you have any advice around someone that has not experienced sort of the enhanced effect of THC and what they should do when they've decided to ingest THC and how to handle that unfamiliar experience?
I think that taking it before bed and taking it at the smallest possible dose available to you, and then only increasing your dose every 4 days, is generally the rule of thumb in clinical practice, so that you can sleep through the altered state. And the altered state will probably induce sleep or support sleep if that's the environment that you are placing yourself in. And if you start low and go slow, you should be okay. And then to optimize your CBD dose, I think that adding THC is certainly phase 2 and should be done with somebody who's got experience using it. But yes, I mean, an unfamiliar altered state can be a very scary place.
Right. Okay. Now we've got, I think, a pharmacological question. Do you know if there is a difference?
I wanted to add to that, if it's still possible.
Yeah, of course.
So you must remember in my presentation, I spoke about what you call an allosteric effect of CBD. So what it does is that if you also take CBD, in response to your last question, if you take CBD, it downregulates the effect of the psychoactive effect. Because of the allosteric effect, it inverses the effect. And that's the advantage of CBD.
Yeah, that's thank you for that. I mean, I guess that's also why maybe some people, certain strains or experiences, there's a lot of agitation, whereas with others, there's maybe less agitation, and that could be the CBD effect, that allosteric effect that you're speaking of. Okay, we've got another question. Do you know if there's a difference in how CBD is absorbed when digested versus when inhaled or smoked? Does the body dissolve the compounds in a different way? Prof?
Yes, completely different way. So in terms of oral digestion, it has to move through the absorption through your intestines, and from intestines it has to go through your hepatic system, your liver, and liver metabolism before it actually gets into your blood or your plasma and getting to the target. So that's the whole process for oral absorption. Whereas if you get inhalation, it almost directly gets into your plasma or your bloodstream.
Excellent, thank you. Okay, I guess we're going to save the best for last. Dr. Sky, I know you spoke about sexual desire. So in terms, we all know what, I mean, a lot of us are very familiar with dopamine. Was it prolactin that you mentioned?
Yes, so what was interesting is that in the group that was studied, the group that after smoking cannabis had an increase in prolactin, had obviously a convert, they had a drop in dopamine, and they didn't have heightened sexual desire. And in the other group, they had a drop in prolactin with a responsive peak in dopamine that did heighten their sexual desire. And their baseline prolactin levels going in sober or prior to the substance had no impact on the outcome. So I mean, we have no drugs for things like this available to us that have any meaningful impact that I'm aware of. So I thought that was just something that was interesting and might be something cool to watch.
Very interesting. Just a quick question on prolactin. What is it, a hormone, or is it? It's not. It's not.
Prolactin is produced in your pituitary, and it's predominantly associated with breastfeeding. It's what causes milk production, but the prolactin is part of your homeostatic function. It's released in men post ejaculation to direct the refractory period. And it's also, like oxytocin, is associated with bonding and connection. So it's quite poetic that post coitus it would hopefully incline a man to cuddle. So it's produced in both men and women, but it's more instrumental in a woman from a feeding of her baby point of view.
All right, the cuddle drug. All right.
Cuddle drug. There we go.
Okay. Well, I want to thank everyone. I want to also thank Nicola for really putting this together. And then, of course, M2Bio for really, was being so instrumental in a lot of the research that we're undertaking. So, Prof, thank you very much for a very informative presentation. Same thing to you, Dr. Sky. And Nicola, I don't know if you want to say a few last words, but
yes, thanks so much. Let me see if I can get my video working again. As you can see, load shedding is hit here. I just want to say thank you so much to Prof Maharaj and Dr. Sky for all of the effort that you've put in for tonight's presentations. It's really appreciated. I know I learned a lot, and I'm sure a lot of people who've attended the webinar have learned a lot as well. So I'd just like to close off with a couple of slides. Let me see if I can share my screen now. Sorry, let me just, it's quite hard to go into presentation mode. How do I do this? Control F5. Okay, sorry. I can't seem to, there's something blocking me here and I cannot see my, oh, here we go. There we go. Okay, cool. So a little bit about M2Bio Sciences for those of you who don't know. M2Bio is a nutraceutical biotechnology company focused on alternative plant-based cannabinoids, medical psilocybin, and mental health therapeutic research powered by artificial intelligence. We also supply evidence-based sustainable products and solutions, which commercializes a range of CBD and mushroom-based products under Dr. Anna RX, Medspresso, and Liviana. So this is a range of the products that we have so far. And then in terms of the research that we're doing, we have partnered with the University of Pretoria and Enterprises University of Pretoria and put together the M2Bio-Discovery Centre for AI-Driven Phytomedicine Research, which is headed up by Prof Maharaj. And a lot of exciting research is going to come out of this space in the future. So keep an eye on that. And in terms of divisions, there's a lot of divisions underneath M2Bio. So we have a sustainability division that's producing sustainable packaging solutions, which is M2Enviro. We have M2 Sentient, which is looking at how the senses can help heal. We've recently launched the M2 MMA division. So we will have that league in future. And then the division that I head up, which is M2 Biome, and it's pertinent that Sky covered how CBD can influence the microbiome. So everything that we do is evidence-based. We'll be producing nutritional products underneath this line powered by AI. So if you have any questions about who we are and you want to know more, feel free to contact us. And again, thank you to everyone for all of your efforts this evening. And with that, I say good night.